Swayam Prabha. Digital India. Educated India.
Okay, so let us get started. So in the last class, we were discussing about allosteric enzymes, and the very last thing was the feedback regulation of allosteric enzymes. In a biochemical pathway, usually the enzyme catalyzing the committed step or one of the earliest steps in the biosynthetic pathway is subject to feedback inhibition by the end product of that particular pathway. So today we'll briefly look at the kinetics of allosteric enzymes, and then we'll go into other ways of regulation of enzymes. So since you have a positive cooperativity kind of situation where binding of substrate to one of the subunits, remember allosteric enzymes are usually multi-subunit enzymes, so that activates binding of substrates to the active sites of other subunits as well. So as a result, the initial substrate binding leads to a significant increase in the rate of the reaction because the binding to the other subunits is increased by this. And as a result, you get this sigmoidal shaped graph. When you plot the, the rate of the reaction against substrate concentration. But still the saturation is the same, so therefore the half Vmax still remains the same. And here, since their behavior diverges from Michaelis-Menten, typical Michaelis-Menten equation where you have that saturation graph. So we don't call it as Km, instead that constant is called K half. Okay, so that's the only main difference, and this important change in the graph. So initially it is not linear. Okay, so in Michaelis-Menten enzyme it would have been linear. So because at the very initial stage the Km is we ignore the concentration of the substrate in the equation, and as a result it becomes Vmax S by Km plus S, and when you ignore S, Vmax by Km remains constant, so therefore the velocity varies linearly with the substrate, right? So in this equation, so we have so V0 equals, you have Vmax S divided by, so in this equation, very early we would ignore this. So then Km by Vmax Vmax by Km remains constant. So V0 equals substrate. So it will vary linearly with substrate concentration. And that situation you don't see in allosteric enzymes, because initial binding increases binding to the other subunits, and as a result, as it binds more, it starts to increase after a lag period. Okay, so this is characteristic shape of the curve for an allosteric enzyme. And when you have a positive interaction, like positive cooperativity, that is binding to one subunit enhances the binding to the other subunits, as we see in oxygen binding to hemoglobin, then it shifts this direction. So this is with a positive regulation, and with negative regulation it shifts in the opposite direction. Okay, so these are characteristics of allosteric regulation. So that is all the kinetics we are going to learn, because we are not going to become enzymologists here. We are just getting a flavor for biochemistry. So this is just to have the basic idea. And many of these allosteric enzymes, in addition to the binding of the molecules, okay, the molecule that binds need not always be substrate. Only in the case of hemoglobin, it is substrate itself. In other situations, I told you the end product of the pathway might regulate the upstream enzyme. So in that case, it is not the substrate, it is another molecule. So it could be any molecule that binds to a catalytic site, regulatory subunit, modifies the overall conformation such that the active site in the catalytic subunit is affected. So remember the R subunit, C subunit cartoon that you saw in the last class. So one of the other ways of regulation of these enzymes is covalent modification. So one of the residues on the enzyme, for example, if this is an enzyme, so it has two subunits, it's phosphorylase. So this is involved in removal of phosphate groups. And when, in this case, the 14th serine, its hydroxyl group, when it gets phosphorylated, it becomes more active. Okay, so in both the subunits, you have two serine residues in two subunits, and when both are phosphorylated, it is active. And when the phosphate groups are removed by phosphorylase phosphatase, so the substrate is this enzyme phosphorylase, and another enzyme phosphatase removes that phosphate group, then it becomes inactive. So by this way, you can have a regulation through covalent modification. So this phosphorylation is a very common modification of many enzymes, variety of enzymes, not just phosphorylase itself. So this phosphorylation based regulation of enzyme activity, when originally discovered, people did not realize its prevalence among a very large number of proteins. And later, when it was realized, this discovery was considered as a very profound, conceptually advancing finding in biochemistry, and the scientists were awarded with Nobel Prize for the discovery of phosphorylation as an important enzymatic regulatory mechanism. And another way of regulation is the enzymes are usually produced as a longer polypeptide than the active form. So a part of the polypeptide, usually the N terminus, gets proteolytically cleaved. For example, chymotrypsin is produced as chymotrypsinogen. So that precursor forms that are inactive are often named by this addition of gen, trypsin, trypsinogen. Okay, and once it is cleaved by an active version of the same enzyme in this particular case, so you have like these 15 amino acids removed, then you have further cleavages, then you have finally the active enzyme trypsin. Okay, so in this case chymotrypsin, and similarly trypsinogen becomes trypsin. And in this case, it's an enteropeptidase, entero referring to intestine. Okay, anything to do with the digestive system you call entero. Okay, that's why you call gastroenterologist, the doctor who specializes in digestion related issues. So trypsin activates chymotrypsinogen by cleaving between this arginine and isoleucine, and then further cleavages by chymotrypsin itself, resulting in the active enzyme. Okay, so this C-terminal portion. And similarly, trypsinogen gets cleaved as shown in this cartoon to become active trypsin. And this sort of proteolytic activity based activation is common in blood clotting pathway as well. And similarly in complement pathway in our immune system. And also in many of the hormones, peptide hormones, they are activated again by proteolytic cleavage. So proteolytic cleavage based activation again is fairly widespread in as a regulatory mechanism. All right, so this is all our discussion on enzymes. So we got a basic idea of what are enzymes and how they achieve rate enhancement. The main point you want to remember there is the various non-covalent interactions that happen that leads to distortion of the substrate reaching the transition state and the enzyme changing itself to fit to the transition state, a phenomenon called induced fit. Okay, and all of the energy, the cumulative energy coming from all these non-covalent interactions, we call binding energy, and this binding energy is the main component of reducing the activation energy. So this is the main concept in enzymes that you need to remember. Then how we relate the rate of the enzymatic reaction, enzyme catalyzed reaction to the substrate concentration using Michaelis-Menten equation and its transformed version, that is the double reciprocal plot. Then you need to remember how the enzyme activity gets affected with various kinds of inhibitors being present. Then the last main concept is the allosteric enzymes. Okay, so if we know these basic concepts of enzymes, then that is good enough for an introductory course in biochemistry. So next we move on to our first major molecules, that is carbohydrates. Okay, so carbohydrates are the most abundant organic molecules on earth, primarily because of their versatility in terms of use, both as structural material like cellulose forms the major structural component, cellulose fibrils of the plant kingdom, and the amount of plants on earth versus animals, and cellulose itself can account for why carbohydrates are most abundant. In addition, they are very useful as storage of energy, like the starch that we primarily depend on for our energy source is carbohydrate. And the exoskeleton of insects, you would have seen many insects, they have their wings are covered by a thick, shiny outer cover. Okay, and those shiny things are made up of a variety of polysaccharide called chitin. Okay, we will see soon. So this chitin and cellulose are extremely abundant. So let us get into the basic structures. We already have familiarized ourselves with the terms like monosaccharides, polysaccharides, aldoses, ketoses, and then glycosidic bond, etc. So we will refresh our memory and get into more details today. So we know carbohydrates, the smallest units are the monosaccharides, which are linked through glycosidic bonds to form disaccharides, oligosaccharides, and then polysaccharides. So first we will see monosaccharides. The smallest is glyceraldehyde, which has an aldehyde group, so it's an aldose, and it has 3 groups, so there are 3 carbons, so we call it as triose. So it's an aldotriose. And you may have instead a keto group, dihydroxyacetone, so ketotriose. So fructose, which is the common ketose that you encounter, like for example, the common sugar that you add to coffee, tea, or milk, whatever, it's a disaccharide. One molecule is glucose, another molecule is fructose. And fructose, the name itself tells you, is very abundant in fruits. Okay, most fruits contain fructose, usually about 4% of the fruit's weight, but some dry fruits, like for example dates, can have way too much fructose to be healthy. So as a result, eating dates, too much of it is not good for health at all, completely contrary to TV advertisements that you frequently see. So the word fructose comes from fruit. Okay, it's rich in fruit. So these two are the most common ones. Glucose, starch breakdown product is glucose, and fructose exists as a monosaccharide in fruits, and in the common sugar, these two exist as disaccharide. So before we go further on to considering the various monosaccharides, let us focus our attention to the stereochemistry a little bit here. So if you look at glyceraldehyde structure, so this carbon is not chiral, it has two same group hydrogen attached. If you take this carbon, one aldehyde group, one hydrogen, and then an hydroxyl group, then you have this, the CH2OH group, this alcohol group, methyl group attached here, okay? And so as a result, these are four different substituents, and as a result, this is a chiral carbon. So in a chiral carbon situations, you can have two different orientations, as we have seen earlier. So the mirror image, this one, like here, the hydrogen and the hydroxyl group project towards you, and they can have an opposite orientation where the mirror images cannot be superimposed. Okay, so two versions therefore are possible, and we usually write them in this form. This is called Fischer projection formula, and we know the perspective diagram that we have learned where the bond orientation is shown, like the solid wedge shape shows that these project away from the paper, the plane in which it is shown, and the dashed wedge means those groups are away from this. Like for example, here if you see in the ball and stick model, this aldehyde group and this the CH2OH, they are backwards and these two are front. And in this, you can rotate and have OH on this side as shown here. And these two orientations, okay, HOH, like in a more familiar way in the written format, I'm going to call on the right side or left side. Okay, it's easy to remember when it is on the left, you say L, which is just for memory, I am saying. These are just conventions. This doesn't mean dextrorotatory or levorotatory. Okay, don't confuse this with the optical rotation, although these are optically active. So when we have this arrangement, like in the way we write, when the OH is on the right side, it is D-glyceraldehyde. When it is on the left side, it is L-glyceraldehyde. Okay. So you can have one chiral carbon, you can have two versions. So the number of stereoisomers possible is 2 power n. Okay, so if you have two carbons, chiral carbons, then 2 power 2. If you have 3, then it is 2 power 3 and so on. Okay, so if we go back and look at it, this has only one. Dihydroxyacetone, no chiral carbon. If you take glucose, you have 1, 2, 3, 4. So as a result, it has 16 different versions possible, and of which the 16 can be separated into two groups. 8 the D version, like HOH in this orientation, and L where this will be on the OHH orientation. So there are 8 names, D, so glucose and so on, like D-glucose, D-galactose, D-mannose, D-iodose and so on, 8 names, and then the same 8 with L. So since it's an introductory course, I'm not going to introduce all those names or expect you to remember all of them. But at least you should be able to draw the structure of D-glucose correctly. Okay. And this DL reference, we use the carbon that is farthest from this aldehyde group. Okay, the orientation of the hydrogen and hydroxyl at the chiral carbon that is farthest from the functional group, this aldehyde group. In this case, this will be the one. fifth carbon. 1, 2, 3, 4, 5, 6. This is the fifth carbon. So in this case, it is this itself. This is the chiral carbon farthest from this, because there is only one, and so on. Okay. So this is the 5-membered aldo sugar, aldo hexose, sorry, aldo pentose. Then you have ribose without the hydroxyl group, deoxyribose. This is in RNA, this is in DNA. Okay. So the next important feature of carbohydrates is their ability to form intramolecular ring structures. Okay, that comes from this reaction. So the aldehyde group can react with an alcohol group, leading to the formation of what we call hemiacetal. Okay, so this carbonyl group gets reduced to hydroxyl group. And since you have another hydroxyl group that can react with another alcohol group like this, then when both are substituted in that manner, you call that as an acetal. So usually the aldehyde group with one of these hydroxyl groups will form a hemiacetal. Okay. And when that forms, you have an additional OH group available. And when that OH group, this one, so this is an intramolecular linkage in a typical aldose like glucose. And the resulting hydroxyl group, when it forms a glycosidic bond with another sugar, so when the OH group it reacts is intermolecular, it is from another sugar molecule, then it forms an acetal. Okay, so glucose linked in sucrose in starch is therefore is an acetal linkage. Okay, similarly ketone with an alcohol can form a hemiketal, and ketal when it reacts with the alcohol group of another molecule. Okay, hydroxyl group from another sugar, it forms ketal. So this reaction is very critical because this is the basis for the ring structure formation. So here you have for glucose this aldehyde group. Most often it is with this fifth carbon's hydroxyl group, and this reaction aldehyde alcohol condensation leads to a ring structure, and this ring structure we usually display in this hexagonal form, which is called the Haworth perspective formula. So this is the Fisher projection formula, and this is the Haworth perspective formula. This is not exactly Haworth. In the perspective formula, we would have used those wedge-shaped bonding to show the projection as well. But this hexagonal ring is usually is referred to as Haworth because Haworth proposed this first. So an important point to consider here is when this ring structure forms, you are generating another chiral center. See, this double bond or this carbon is not a chiral carbon, but once this ring forms, it is a chiral carbon. And this carbon is called the anomeric carbon. Okay, and two sugar molecules, for example, in this case, these two differ only in the orientation of the substituents of this carbon. All other groups, the orientation of those groups on these carbons are all same. Only here it differs. And the sugar molecules that differ in this fashion are called anomers. And this carbon itself is called the anomeric carbon. So in aqueous solution, this like one orientation we call alpha and the other orientation we call beta. By convention, when you show the OH on the top, you call the beta, and below it is called alpha. So in starch, what you have is an alpha D glucopyranose. Okay. So orientation on this carbon is the reason for D, and orientation around this anomeric carbon is the reason for this alpha. So the normal glucose is alpha D glucose. Okay, this pyran I'll come in a minute why we use this word pyran as well. So the main point is this intramolecular hemiacetal formation results in the generation of additional chiral center, and that chiral carbon is the anomeric carbon, and when two molecules differ in the orientation of the substituents only in the anomeric carbon, they are the anomers. And in solution, one form rotates to the other form readily, and they interconvert readily, and that process we call mutarotation. These do rotate the plane polarized light. So the dextro and levo rotatory comes from this carbon. And in solution, they exist in a certain equilibrium. I think this is more than this. You need not worry about that detail. But the conversion of one optically active form to the other optically active form with respect to anomeric carbon is what is mutarotation. Okay. And in this fashion, this ring structure resembles the organic molecule pyran, and therefore we call them as pyranos. So the proper chemical name for glucose is alpha D glucopyranose. So now you know why is it alpha, why is it D, and why it is pyran. And similarly, the keto sugars, they resemble furan, and therefore they are furanose. So alpha D fructofuranose. D comes from the fifth carbon, and alpha is from the first carbon that formed the hemiketal structure here. So this is the basis for the way we for the nomenclature of the monosaccharides. So now we look at some of the derivatives of the monosaccharides that are really important. So this is the first one, glucose, we know very well. So this is the most common one. So here it is shown in the beta form, OH on the top. Okay. So the naturally occurring sugars are mostly D version. So therefore we are going to, for our convenience, ignore the L versions. So all of them are going to be D. So it's D glucose. So here this diagram is beta glucose. So quite often the second hydroxyl group is substituted with an amino group. So they are called glucosamines. And these amines are often substituted with an acetyl group. So because it's an amino group that is substituted with acetyl group, we call this N-acetyl glucosamine. So these are usually present attached to lipids or proteins in our system as glycoproteins or glycolipids, and they become important functionally in many different situations. So we will consider them when we get to those actual contexts. So these are very prevalent. And in addition, you may have phosphate groups. These phosphorylated versions are very important because the phosphorylated versions do not readily get out of the cells. So it helps in trapping the monosaccharides in the cytoplasm because there are no transporters for this phosphate version, for phosphorylated versions. And second, this is an activated bond here compared to this hydroxyl group, the alcohol group. This phosphorylated version is more reactive. So this is an energetic form which is a good substrate for further reactions. Okay, for example, formation of polymer like glycogen in the liver. So you have phosphorylated version. Then in addition to this amino group as well as this acetylated amino group, sometimes you have an R group in the adjacent carbon, the third carbon, and that R group is usually lactic acid. Okay, and this lactic acid substituted and then this lactic acid substituted version of glucosamine. So this structure is glucosamine with the lactic acid. We call that as muramic acid. So this muramic acid also might have acetyl group, N-acetyl muramic acid. So this is a very important component of bacterial cell wall. We will very soon see that. And these are some of the modified versions. Basically, the major modification is the amino group, which gets derivatized with acetyl group. And sometimes you might have a lactic acid attached as well. And so this is other, the galactosamine, manosamine, like that. And some of the sugars might have a methyl group instead of a hydroxyl group in the 5th position. Fucose, rhamnose are examples. They are from mannose and galactose respectively. Then you may have a situation where the aldehyde group here, this anomeric carbon, may be oxidized to a carboxylic acid form. And they are called as aldonic acids. So glucose is like gluconic acid or gluconate in the ionized form. And they usually react with this alcohol group and form ring structure called glucanolactones. Some of them may have similar oxidized form in the CH2OH 6th carbon, and they are called uronic acid. In this case, it is glucuronic acid. Okay, aldonic acid or alduronic acid. Aldonic acid means the 1st carbon. Alduronic acid means it is the 6th carbon that is converted into carboxyl groups. So in addition to these common derivatives, one other important components that we often encounter in our system, where usually found in glycoproteins, is this N-acetyl nuraminic acid. This nuraminic, do not confuse with muramic. This is muramic, this is nuraminic. This is 6-member, this is a hexose derivative, while this is a 9 carbon. So the R group has 3 here. And they usually have acidic group as well as an N-acetyl substitution as well. The common name is sialic acid, and this is a very important modification of glycoproteins required as address tags. We will consider it towards the end of this chapter. So these are some of the derivatives of the common hexoses that we are familiar with. So a third important property of carbohydrates is their ability to reduce potential oxidants. For example, cupric ion can be converted into cuprous by this aldehyde group. So this aldehyde group, remember this hemiacetal is always in equilibrium with this unlinked form. So let me go back to it to highlight that point. So this version, hemiacetal, is in equilibrium with this aldehyde version. So if you are going to draw this away through another reaction, then it is going to tend towards this to balance and reach equilibrium, and therefore more and more of the cyclical form, the intramolecular linkage is cyclical, and that will get linearized and used up in the reaction. So in this version, do not ignore its ability to behave like an aldehyde. And its ability to behave like an aldehyde is because of this equilibrium. And so due to this, so this one is in equilibrium with this linear form. And in the linear form, this acts as a reducing agent. And such sugars are called reducing sugars. Okay. So in a long chain of alpha-1,4 linkage of glucose, which is the starch or glycogen. So there, one end of the chain will have a glucose molecule like this. Its 4th carbon is linked, but the 1st carbon is anomeric carbon is not linked in glycosidic bond. So therefore, this can be in equilibrium with this aldehyde form and this can reduce. So that end of the amylose chain that has this free anomeric carbon, which is free in the sense it's not engaged in glycosidic linkage, can act as the reducing end. Okay, so that's the convention for why it is called reducing end. And this ability to reduce is the basis for a test called Felling's test. So here the cupric ion becomes cuprous, and that in alkaline condition can form a reddish precipitate. This was the basis for detecting glucose in the blood and used as a diagnosis for diabetes. So you may not have seen anyone doing this, but when I was a kid in school age, most of the grandparents, when they were diabetic and they were asked to do the glucose measurement in their urine at home itself, they will do this test. So it's standardized, ready-made thing available, and they will do it and they'll find it is the solution itself will be blue. If it is greenish, means they are okay, but if it is reddish brown, then the sugar level is high. But nowadays we don't do that. We do a lot simpler and more accurate one, which is based on this enzyme called the glucose oxidase. So which forms this gluconolactone. Remember this structure. Okay. When the aldehyde group is oxidized to carboxyl group, and when that readily forms this ring, this ester linkage, carboxylic acid with this alcohol hydroxyl group, that is the gluconolactone. And that is what glucose oxidase does using oxygen as the other substrate. You get the gluconolactone. The other product formed in this is hydrogen peroxide, which can be converted with the help of another enzyme, and that that reaction can result in colored product, and that is what we measure using peroxidase enzyme. And remember I have told you about catalase and peroxidase when we were talking about the turnover number. So using that enzyme, we make colored product, and since that enzyme has an extremely high turnover number, it's extremely sensitive. So very small amount of glucose also can be measured using these two enzyme coupled reactions. So essentially for measurement, the end product has to be colored. Therefore, you can use a simple colorimeter where absorbance is based on the concentration of the given colored substance, and we calculate the amount of glucose present. So it is all because of this reducing end, this aldehyde group. Okay, so now we move on from monosaccharides to disaccharides. Okay, so again remember the monosaccharides, we considered aldoses and hexoses. When I say those two names, remember the functional group aldehyde and ketone. Then the other important thing we learned is hemiacetal hemiketal formation, that resulting in additional center of chirality. So anomeric carbon. So these are important concepts. Then the common derivatives of amino group and N-acetyl derivative amino group and so on. So the next one we are going to look at the disaccharides. So two glucose molecules in starch itself or cellulose itself is a disaccharide. So you can consider them as a disaccharide repeating unit. Apart from that, there are carbohydrates that exist as disaccharides as such. For example, in milk, we have beta-D galactose. Like this hydroxyl group is shown above the ring, and therefore it is beta. So beta-D galactopyranose is in glycosidic bond with glucopyranose. That is again beta. So that beta-1,4 linkage between galactose and glucose is lactose. This is the primary sugar present in milk, lactose. Okay. And here you have a reducing end here. So therefore, lactose is a reducing sugar. And one other important detail I want to tell is when two sugar molecules, like for example, if you take glucose and another molecule where they differ only in the orientation around one of the chiral centers, they are called epimers. Okay, glucose and galactose are epimers. So we have learned enantiomers, diastereoisomers, anomers, and epimers. Enantiomer, mirror images. Diastereoisomer, the same structure, but the functional groups are attached to two different carbons such that they are not mirror images. Therefore, they are diastereoisomers. If they are mirror images, there are enantiomers. And when the orientation differs only around anomeric carbon, they are anomers. If the orientation any of the one single chiral center, they are epimers. Okay. Sucrose, on the other hand, has an anomeric carbon and another anomeric carbon. Okay, so here we have inverted the structure. So fifth carbon is here, the first carbon, anomeric carbon is here of fructose. So 1, 2, and this is beta orientation, and this is alpha orientation, this glucose. When you have glucose fructose joined like this is sucrose. So here both the anomeric carbons are engaged in the acetal formation. So this is acetal. So this structure is analogous to this. Okay. So since we are talking about one of them ketose, it is analogous to this structure. So this itself is, if you consider as the internal ring, this is like this R is the glucose, and this whole thing is the fructose in sucrose. And in such a structure, you have no reducing group available, and therefore this is non-reducing sugar. Similar is the case with the trachalose, which is glucose-glucose alpha 1,1 linkage. This is a disaccharide present in the circulating fluid, like the blood equivalent in insects, and this is important source of energy for insects. So these are the common disaccharides that we encounter. So when you take starch and convert into disaccharide like glucose alpha 1 for glucose, it's called maltose, and maltose is often present in some of the cereals, and so that is where you have this malt in some nutritional supplements. They are more readily digestible than large starch molecules. And then finally we move on to polysaccharides. Polysaccharides are quite simple, the long chains of monosaccharides, and we already saw alpha 1,4 in amylose part of starch, and we saw the branches alpha 1,6 branch in amylopectin of starch. So starch therefore has amylose and amylopectin, unbranched and branched versions, and glycogen is more like amylopectin but with a lot more branches than amylopectin. So this is the common storage carbohydrate in animals, particularly in liver is where it is stored. So then you may have these are homopolysaccharides because this all monosaccharides are the same like glucose. So when you have different monosaccharides are joined, they are called heteropolysaccharides. So you may have two alternating ones and you have multiple ones and so on. Okay. So we look at structure of some of them, like for example, one of them we need to look at chitin because we are familiar with starch, glycogen, cellulose. Cellulose is glucose but beta 1,4 linkage. So chitin we'll look at it. So chitin is beta 1,4. So the hydroxyl group is above, so therefore you show like this. Okay. Instead of showing it down. And this one has an N-acetyl group. So it is N-acetyl glucosamine. So this group is N-acetyl glucosamine. in beta 1,4 linkage with N-acetyl glucosamine, and therefore it is beta 1,4. This disaccharide keeps repeating. So beta N-acetyl glucosamine repeat is what is chitin, and that is what is this shiny exoskeleton of insects. Okay, so this chitin, the disaccharide is called chitobiose, and this chitin is the second most abundant organic molecule on earth, only behind cellulose. So you may have seen, insects are very large numbers. Not only insects or other arthropods like shrimp, prawn, etc. Their exoskeleton also contains chitin. And the chitin molecules are used as biomaterials in tissue engineering as well. So understanding their chemistry is very useful for many applications too. So the third is heteropolysaccharides. So what I'll do is I'll stop here. We will consider heteropolysaccharides and other derivatized form of polysaccharides in the next class. So far we had minimum derivatization. This N-acetyl amino group is the only addition we have seen. So we are going to see a lot more complex versions, and they are very important in our extracellular matrix. So we will take them up tomorrow.